As we learned last week, eating a high-fat diet can rapidly alter insulin sensitivity and glucose uptake in skeletal muscle. As the figures show, blood FFA content was elevated following a high-fat diet, and skeletal membrane GLUT4 content was reduced. Today, we will examine a potential mechanism that may explain the link between elevated blood FFA and reduced membrane GLUT4 following a 48-hour high-fat diet.
Let's first start off by creating a center of mass model. So our mass will be skeletal muscle GLUT4. The inflow will be insulin signaling, which will stimulate translocation of GLUT4 into the membrane. And the outflow will be the reuptake of GLUT4 into glucose storage vesicles. Insulin present in the blood will bind to the IR receptor, activating IRS, PI3K, and AKT. PI3K activates RAC, and AKT will phosphorylate AS160, inhibiting it and allowing translocation of GLUT4 into the membrane and allowing for glucose to enter the muscle. As presented in the question, a high-fat diet will result in an increase in blood plasma fatty acids. This will then result in more fatty acids in the muscle, which can then go through two separate pathways. It can go through the reacidification process, increasing DAGs and therefore inhibiting IRS, and it can also go through the sphingolipid formation and increase ceramides and inhibiting AKT. This will then result in less AS160 being inhibited and remain on the GLUT4 storage vesicles, resulting in less GLUT4 on the membrane. We hypothesize that an accumulation of ceramides will inhibit AKT and therefore suppress insulin signaling. We propose that this is the reason for the difference in skeletal muscle membrane GLUT4 content in the placebo and in the high-fat diet group. So as an overview, a reduction in insulin signaling will result in a decrease inflow into our mass. This will then result in a decrease in skeletal muscle membrane GLUT4. This current study has demonstrated that a high-fat diet, which includes saturated fats, increases intramyocellular content of ceramides, reinforcing our hypothetical association between high-fat and ceramide content. This study also demonstrated that AKT phosphorylation, a key component of the insulin signaling pathway, was reduced in the presence of ceramides and insulin compared to when ceramide inhibitors and insulin were present, as shown in Figure A above. The previous study demonstrated the association between a high-fat diet and the inhibition of AKT. This study will look at the association between the inhibition of AKT and GLUT4 translocation. This study's primary objective was to determine the influence of AKT on AS160 phosphorylation. They measured the total AS160 and phosphorylation was detected in AKT substrate. They injected 10 nanomoles a liter of Wortman, a PI3K inhibitor, precursor to AKT activation, into the extensor digitorum longus on mice. This graph shows that an inhibition of AKT resulted in a reduced inhibition of AS160 phosphorylation. The study showed that these results directly relate to a reduced GLUT4 translocation into the membrane.
This research demonstrates that ceramides are elevated following a high-fat diet, resulting in reduced insulin signaling, decreasing the inflow of our mass model. This increase in ceramides led to a greater inhibition of AKT, which reduced the translocation of GLUT4 to the muscle membrane. These results support our hypothesis explaining the link between eating a high-fat diet and the resultant decrease in skeletal muscle membrane GLUT4 content.